we have already seen the tandem MS of a peptide, and the sequence of the peptide is GPFNA, and we know that G stands for glycine, P for proline, F for phenylalanine, N for glutamine, and A for alanine. Now we will see the protein sequence and identification of this peptide, and we can see that the theoretical spectrum TP of peptide P can be calculated by subtracting all possible ion types, that is delta I to delta K, from the masses of all practical partial peptides of P. That generates the K masses in the theoretical spectrum. Here we can see that the peptide that we called it GPFNA, when it is breaks up and generates small peptides, then we can see that the small molecules that we call them the ions, they release from these peptides, and the two are the most important ions. One is the water molecule and the second is the ammonia molecule. And we can see that the sequence of this peptide and the mass of this peptide is 498, and we know that the mass of water is 18, and if we minus 18 from 498, it comes out to be 480. And similarly, we know that the mass of the ammonia is 17, and if we subtract 17 from 498, it comes out to be 481. And similarly, we can calculate for all of the peptides different sizes of peptides that are generated in this mass spectrophotometer. And by subtracting the value of water molecules and the mass of the ammonia, we can calculate the size and mass of all the peptides. So in the experimental spectrum, S starting from S1 to SQ is a set of numbers, and we can see that the difference between the theoretical spectrum TP and the experimental spectrum S is that TP can be generated mathematically for a given peptide, that is a P, but the spectrum S can be experimentally generated without knowing the peptide sequence, as we know earlier. The match between the experimentally generated spectrum S and the peptide P is the number of masses in the spectrum S. That is equal to the masses in the theoretical spectrum, that is TP. And it is also known as the shared peak counts. And we know that the algorithm that we use for the calculation of and the identification of peptides is the algorithm that uses the more sophisticated functions as compared to the shared peak counts. And because we use the very specific algorithm that use the more specific functions, and it on the other hand also give the specific weights to the spectrum and the peptide. We can formulate the peptide sequencing problem as follows, and we know that we want to find a peptide whose theoretical spectrum has a maximum match to Mayer experimental spectrum, as we have seen in the previous slides. So the input of this algorithm will be the experimental spectrum S, that we will do some mass spectrophotometry and we will find the spectrum S. The set of possible ions types that we have seen, that it could be water and ammonia and maybe some other ion types, and the parent mass M. We can also calculate the mass of the parent peptide. So these are the inputs that we will provide to the algorithm, and as the output we will get a peptide P of mass M whose theoretical spectrum matches S better than any other peptide of mass M. So by comparing and matching the mass of a peptide, we can get the correct sequence of the mass, that has the peptide mass.